So last week we did talk about biomolecules some, but this week I want to start at carbs and go all the way through to proteins. We haven't gotten into nucleic acids yet, but I do have slides on that. So let's go ahead. This is the same PowerPoint as last week. This is kind of the same material. So let's start with carbohydrates. So carbs are used for fuel and building blocks, and this is kind of intuitive. Fuel, this is why they tell athletes to eat pasta before a game or something of that sort. Let me get this up. Building blocks, they can be used to build a structure. Now, how to identify it, you guys, the number one thing I think from this chapter is you need to be able to identify a molecule when it is given to you. So how you would identify a carb is a carbonyl group. So this C double bonded to an oxygen right here. But another thing that you can try, that you can identify carbs from, let me change colors here, is the amount of OH groups. Because you can see this C double bonded to an oxygen very well in the chain structure, but not the ring structure. It's just kind of, that's not it. It's just kind of hanging off there. So what you can see in the ring structure is OH, 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 OH. You can see a lot of those hydroxyl groups. Okay, did I hear a notification? I'm hearing stuff. Okay, just to make sure, so everybody can hear me and see my screen, right? Make sure I'm not talking to myself quite literally. Yes, thank you. Okay. So a lot of OH groups. That's the one major way to identify a carbohydrate. Now, dehydration synthesis, I kind of broke this down for you in kind of a equation. So for dehydration synthesis, you have 2 sugars. You take out a water, you get 1 big sugar. Hydrolysis, you have 1 big sugar. You add water, you have 2 sugars. Okay? Now, remember, it is easier when looking at this if the Janet and Carl analogy worked for you. That's great. But I just want to give you all another way of looking at it, and that is just counting the molecules. So here we have a polysaccharide, or sorry, a disaccharide, because it technically only has 2. Oh, that jumped. Why did that jump so much? Okay. So here we have a disaccharide. I'm positive I'm not spelling this right. Okay. Now we're going to add water. Okay, so we have H2O. And then what's going to happen is this bond is going to break. So then we have this with this oxygen, but this oxygen needs a hydrogen, so it'll be like that. And then I'm going to bring this guy down here, and he doesn't have anything. So then we're going to add the oxygen from here, and then the other hydrogen. So the main thing to note is, especially for carbohydrates, when you are doing dehydration synthesis or hydrolysis, you are focusing on the hydroxyl groups. That's what you're focusing on. Okay? Okay. All right, so carbohydrates have a lot of OH groups, which makes it polar. So the main thing when we say why is it doing that? The main thing when we say polar is that it has a lot of oxygen. That's what makes something polar. So water is polar. Oh, hydroxyl groups are polar. So that means they mix well together. They both play well. And if something mixes well with water, then it is hydrophilic. Now, remember to break these words down. Hydro means water. Philic means like. So this word literally means likes water. Okay? Okay, so lipids. On the flip side of this, let me switch colors again here. Lipids have a lot of hydrocarbons, which is what I have here, is just a carbon with a lot of hydrogens attached to it. It does not have a lot of oxygen. Why is it doing that? It does not have a lot of oxygen. Now, it does have a carboxyl group, which is this right here. A C with an O, a double bonded O. That is called a carbonyl. Now, when you add a why does it keep, what are you doing? So sorry, guys. I don't know what's happening. All right, so you have a carbonyl right here, and then you have a hydroxyl. So carbonyl plus hydroxyl, you kind of combine those two words together, is carboxyl. Okay? Now, the main thing is when you're identifying these lipids, you need to make sure that I've seen a lot of kids just be like, look at this. Let me erase this. Look at this lipid and say, oh, here's an oxygen, that's a carb. That's not the case. Every single lipid is going to at least have 2 oxygens because every single lipid has at least 1 carboxyl. You need to look at how many oxygens to carbons. So here we have 2 oxygens to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 carbons. That's a very small ratio. So that's how you would determine that it is in fact a lipid. Now, lipids in water, so it has a lot of hydrocarbons, very little oxygen, which makes it non-polar. So just remember, water is polar. So I think of this, the water is the nice kid. It likes to play well with others. Lipids are the non-polar. They don't play well with others. They're isolated, they're mean, they're like the grumpy kids on the playground. Now, if you have one that's very cheery, so let's make water very happy, and then one that's very not, they're not going to mix well with one another. Okay, that's how, then you break this word up. Hydro is water, phobic, phobia, fearing. Okay. Now, types of lipids, you have fats or triglycerides. That's what fats make up triglycerides. And then phospholipids. Remember, phospholipids make up the cell membrane. And then steroids, which are your ring structure. Okay? Okay, so fatty acids. Remember, fatty acids, I'm going to put up here, they join to form triglycerides. Okay? So a triglyceride is 3 fatty acids joined together by ester linkage. So we need to make sure we know the bonds that hold each type of molecule together. So for lipids, most of the time it's going to be ester linkage. For carbs, it's covalent bonding. And then I'll get down to proteins. So there are two major types of fatty acids. One is saturated, which just means that every single carbon is bonded to a hydrogen as many times as it can. So it's kind of straight. It's very pretty looking. Unsaturated is where you have at least one carbon that is not bound to as many hydrogens as it can be, so it has a double bond. All right, so this double bond makes a bend. Okay, so unsaturated, why does this keep what is happening? Okay, sweet Jesus. All right, so unsaturated kind of means, like, I think of broken, because it looks kind of broken. It doesn't look straight. It doesn't look pretty like the other one does. Okay, now we're going to move on to phospholipid. I will tell you all, I haven't looked at the revised test yet. Also, reminder, your test opens next Friday, not tomorrow, but the Friday after that. So guys, please start reviewing this material. Please, please, please. And I will tell you, on the exam, she loves phospholipids. Okay? I've been her TA for 4 years. She loves phospholipids. So make sure you know this, okay? All right, so a phospholipid has a polar head, and that is due to phosphate group. It has nonpolar tails, which are 2 fatty acids. Now, the 2 fatty acids, you have one saturated, so one non-broken one, and one unsaturated, so one broken one. Okay? And then these are the major things it contains: fatty acid, a glycerol, and a phosphate group. And you also really, really, really need to know that phospholipids make up the cell membrane. So let's draw this out. You know that the phospholipids have the polar tail, the polar heads. So they line up outward facing. And then they have their little tails tucked in. Okay, now this is inside of the membrane, so inside. This would be like the environment. Let me see if I can write. Okay, and then this would be inside the cell. Okay, so these are facing out on either side of the cell. This layer is surrounding the inside of the cell, and we know, and we will learn, that the cell is made up of like a gooey substance called the cytoplasm, which is made of water. So we definitely don't want the part of the cell membrane that touches that gooey substance to repel water. And then the environment, we know that most of Earth is water. So if you are a bacteria or a human cell or whatever you are, this side of the cell is going to come in contact with some kind of water. Okay? All right, in steroids, I know when we think of lipids, we usually think of a ring, blah, blah, blah. We usually think of a linear structure, but steroids are rings. Now, you might look at this and be like, oh, here's an oxygen, here's an oxygen, 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 oxygen. Oh my goodness, it's a carb. But remember, each hair mark is a carbon. So I can tell you that most of these like equal rings have 6 carbon. So 6, 12, 18, this has 5 carbons. So there are 23 carbons to 1, 2, 3, 4, 5 oxygens. See, that's still a very, very low ratio. Okay? And steroids are so important in the body because they carry important messages. So like testosterone, that's a steroid. Things like that. And remember, lipids don't dissolve in water. And most of your blood is made up of plasma, which is mostly water. So when we think of this, if we wanted to send a message to the brain, we don't want it to dissolve in our blood halfway there. Then nothing would ever get through to our brain, and nothing would really come of those messages. Okay, any questions about lipids or carbs before I switch over to proteins? Okay. All right, proteins, most versatile. They can be used as enzymes. We will very much talk about enzymes next unit. They can also be used for structures. Enzymes is probably the most common thing that you will hear them do in this class. So every protein has this carboxyl group. And we have seen a carboxyl group before. A lipid also has a carboxyl group. So that might not be the best identification tool. Now we have this R group, which I know she showed in class all of the, that looks too similar. Let me change. She showed us in class all the different R groups. I want to reiterate what she said. You will not have to know how they interact with each other, what each R group is named. You don't have to know any of that. I was at a 400 level biochemistry class and he still didn't make us memorize the R group. So you guys are good. You don't have to memorize them. Just know that they are there. And the R group is what differs. Because every single protein is going to have a carboxyl group, an amino group, which we'll get to in a second, and this hydrogen. What differs is the R group. Okay. Now we get to this amino group, and we have a brand new molecule here that we have never heard of, this nitrogen. So what I will tell you, and this will become very clear when we get to the nucleic acids, because nucleic acids also have nitrogen, but they're not in this little amino group. So more than likely, if you see a nitrogen attached to a carbon directly, then you're talking about a protein. So an amino group is just a nitrogen with 2 hydrogens attached to a carbon. Okay, so it's very important that you know the overall structure of a protein or of an amino acid. I should also say that these are amino acids equals, amino acid is a monomer of a protein. Is the smallest unit. This would be an amino acid. Okay? So this whole thing would be an amino acid. All right, now we're going to move on to the levels of organization of a protein. You will need to know the bonds to every single level. So I have some mnemonics for you. You will also need to know that tertiary and quaternary. I know we haven't talked about quaternary yet, so I'll kind of introduce it to you all. Why does it keep doing that? These two are functional proteins. So functional proteins because they are 3D. Okay? All right, so primary are held together by peptide bonds. Primary peptide, the P's go together. And they're flat. They're just a flat sequence. All right, primary peptide. All right, secondary, that, my friend, is the one that I do not have a mnemonic for the bonds. Probably just going to have to memorize that. And they are held by hydrogen bonds. So remember, these are the alpha helixes, which are the kind of swirls. And the beta pleated sheets, which are like the kinks. Now, it's very, very important to know that these are still 2D. They are kind of kinked and bent, but they're 2D. So they're not functional yet. I didn't leave my room and I didn't leave enough space. Okay. Not yet functional. There we go. That's functional. That's an L. All right. Now, tertiary, I'm going to tell you a mnemonic that you will always remember. Tertiary is held by side chain interactions. You will not need to know what type of side chain interactions. If it's hydrogen bonds, ionic, blah, 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 blah. You just need to know side chain interactions. So how I remember this is Terry is a side chick. All right. Tertiary is a side chick, side chain. Don't mean to offend any of the Terrys out there, but it works. I promise it works, okay? Terry is a side chick. So tertiary is held together by side chain interactions, and this is the first level of 3D, which means this is where it begins to add function. Okay, now quaternary, I know we haven't talked about this yet, so I'm just kind of going to briefly graze over it. Quaternary is multiple polypeptide chains. So I could also say multiple, oh my gosh, multiple tertiary sequences. So imagine tertiary as one ball of headphone, one set of headphones like balled up together. That's a tertiary. Now imagine quaternary if you put like 3 different set of headphones and they all get tangled up together. Or if you take 3 different pieces of paper and you crumble them up. They're different subunits that you can kind of pick out. And that's what a quaternary is. It is, you can also think of this as like a complex protein. Oh my gosh. I have to be like hitting something with my arm to make it do this. Please excuse me. Okay. Quaternary is complex protein. And they are held by ionic or hydrogen bonds. So how I remember this is if I had a quarter for every high I heard. If I had a quarter, quaternary, for every high, hydrogen or ionic HI. If you guys have any other cool ways to memorize quaternary, because I know that's kind of a hard one to memorize, or if anybody comes up with a good one for secondary, please let me know, because I cannot find a good mnemonic for secondary means hydrogen bonds. Okay, so I do just kind of want to glance over nucleic acids, because the next study session I will be having is for the exam, and I know for a fact we will get through nucleic acids tomorrow. So I do kind of want to glance over it just a little bit. So a nucleic acid contains a 5-carbon sugar. So you have this right here. And then it contains a nitrogenous base. This right here. So this is where ATCG comes in, if you guys have heard that with DNA sequencing. And then a phosphate group. So we have heard of phosphate before. What also had a phosphate group? What major structure have we talked about that also had a phosphate group? A phospholipid, right? So phospholipid also have. Okay? All right, now, there are 2 major types of nucleic acid. We have DNA, which is double-stranded. You do need to know DNA is double-stranded, so just remember the 2 Ds. DNA is double, and it is long-term storage, and RNA is single-stranded, so it's just the opposite. And this actually goes out and does stuff. So I don't want to get too much into the different types. She'll definitely talk about that more in class tomorrow, but I did just kind of want to introduce it to you all. Okay? All right, so that's all I have. I know I just really wanted to deep dive into, let me stop sharing my screen, deep dive into biomolecules. What I want to do now is I want to open the floor for questions about research lesson 3. First things first, have everybody at least started looking for a topic on research lesson 3? Have you kind of had any issues? Does anybody have any major questions about research lesson 3? Okay, well, I'm not hearing any. I am going to go ahead and let you go. Let me make sure that I take a photo so I can still take attendance. Okay. Okay. And then I'm going to stop the recording. Thank you guys so much for coming. You all are free to go, but I will hang around for a little bit. So if you guys have any questions you want to ask me one-on-one, I'll still be here. Please, guys, make a tutoring appointment if you need extra help. I would also like to mention that tutoring appointments must be made 48 hours in advance. It's just how our system works. So if you want an appointment for research lesson 3, that's due Sunday, you're going to have to book it by tonight or tomorrow morning at the latest. Okay. Also, please feel free to email me. I'm on the Remind app. If you want to shoot me a message through Remind, that's perfectly fine. Yeah, thank you all for coming and have a great evening.